The Anatomy Coloring Book by Wynn Caput and Lawrence M. Elson. The Anatomy Coloring Book. And I'm just going to flip through these pages here. And we are on plate 3. Organization of the body, cell division, mitosis. And I'm going to go ahead and rip this out of here. Struggling, but come on. Okay, there we go. All right. Zoom in a little bit. Okay, cell division. And mitosis. I'm going to color that in. A vital characteristic of all living things is the ability to reproduce its kind. Living things are composed of cells, and it is cells that reproduce in a process of duplication and division called mitosis. Some cells reproduce regularly and frequently, epithelial and connective tissue cells, and others experience division only under specific circumstances, if at all, nerve cells. Failure of certain cells to grow and divide generally constitutes atrophy without growth. Uncontrolled mitosis constitutes cancer. And I'm going to go ahead and color in these words: cell membrane, nuclear membrane, nucleus, chromatin, chromosomes, chromatids, chromosomes, chromatin, chromatids, chromosomes, chromatin, centromeres, kinocatures, centrioles, asters, spindle fibers. I may have said that wrong. Kinetochore. It's kinetochore. Okay. But I'm going to color the nuclear membrane orange. The nucleus will be this brownish color. So this is chromatin D star, and I'm going to color it a lighter green. And this is chromosomes D1 star, and that will be a darker green. chromatids, I will color pink, and that is lettered E, since these pictures are color by letters. E for chromatids in pretty pink. And chromosomes E1, I'll do in this magenta color. For chromatin E2, I'll do in this lighter pink color. And chromatin D star compared to chromatin E2 are at different levels, so they're different colors. Chromatids F, I will color in this blue shade. And chromosomes F1, I will color in blue in another shade, contrast. And chromatin, for the third time, F2, I will also color in blue, just a different shade, 3 shades. And centromeres, the term kinetochore, G, that will be colored in this pretty sea green blue color. And again, that term is kinetochore. Kinetochore. And it's a complex of proteins associated with the centromere of a chromosome during cell division, to which the microtubules of the spindle attach. Kinetochore. Well, it's kinetochores because this is plural. There's an S at the end, so kinetochores. Kinetochore. Kinetochores. And centrioles, I will color red, 1 shade. aster's eye, that will be a shade of orange. And the spindle fibers, that will be this pretty green-blue color. And that is lettered J. All right, so in that yellowy color, I'll be coloring the cell membrane, not coloring all over that circle right there. Cell membrane, yellow. And then coloring the nuclear membrane, the orange color. Coloring in that nucleus. And the chromatin here is the D star, and I'll color that the light green color. Just coloring in that chromatin. And now the centrioles. Interphase. The drawings primarily reflect changes in the nucleus because the significance of mitosis lies in the duplication and subsequent division of DNA, the genetic material. Mitosis usually occurs rapidly within minutes. The period between successive divisions is called interphase. It is during this period that the DNA in chromatin is doubled preparatory for the next mitosis. The observed nuclear changes during cell division are described by phases. And right now I'm coloring in that cell membrane. And the nucleus. But in this orange, I'm coloring in that cell, the nuclear membrane. although I colored it in this light green, I should have colored it in the darker green, D1. It's actually chromosomes, not chromatids. Why did I move the camera? I don't know. My bad, y'all. Okay. I don't know why I just colored piece of the centriole either, but I'm coloring the spindle fibers, and right now I am coloring the asters. Pretty orange color. Aster, asters, asters. And like I said, that's the chromosomes. Nuclear membrane. Nuclear membrane. There we go, finishing up. And coloring the other centrioles. And coloring the spindle fibers. And also coloring the chromatids and, well, both chromatids, they're in different colors because they're different chromatids. And now we're on prophase. Prophase, dispersed chromatin, B star, begins to thicken, shorten, and coil, forming condensed chromatin or chromosomes. D1 star. There are 46 of them. Each chromosome, only 4 are shown for simplification, consists of 2 chromatids, E and F, connected by a centromere, G. Each chromatid has the equivalent DNA of a chromosome and will be called a chromosome in anaphase, as you will see. Note in your coloring that one chromatid, E, of each of the 4 chromosomes is destined for one daughter cell, and one, F, is destined for the other daughter cell. As prophase ends, the nucleolus disappears and the nuclear membrane dissolves. In the cytoplasm, 2 pairs of centrioles, having duplicated an interphase, projects asters of microtubules. The pairs head for opposite poles of the dividing cell. And right now I'm coloring in the centriole. Asters. More asters. And these chromatids, I'm going to color in pink. And these other chromatids, I'm going to color in blue. And I'll go ahead and color those spindle fibers. And now we're on metaphase. Metaphase. Strands of microtubules, spindle fibers, project across the cell center from one pair of centrioles to the other. 46 pairs of chromatids and their centromeres, 4 shown here, begin to group on the spindle fibers in the cell center. And I'll go ahead and color this cell membrane. And I'll go ahead and color these chromosomes, F1. So they went from chromatids to chromosomes. And the same for E, the pinker pink. And then the centrioles. Can't forget those spindle fibers. So ever so quickly, anaphase, the centromeres divide, each daughter centromere attached to one of the 2 chromatids. The chromatids are no longer paired. They are rightfully called chromosomes now, E1, F1, and there are 46 of them being drawn to each pole of the dividing cell by their centromere. 4 are shown here, going to each of the 2 poles. Anaphase ends when the new daughter chromosomes arrive at their respective poles. Telophase. Telophase, the cytoplasm begins to cleave, pinching the dividing cell into 2 new cells. Each new cell, the nucleus and nuclear membrane, are reconstituted. The chromosomes begin to disperse as the centromeres disappear. The cytoplasmic organelles are segregated into the 2 daughter cells as the cleavage of the cytoplasm is rapidly completed. The daughter cells, like their mother cell before them, will remain in interphase until their time arrives. So I'll go ahead and color this cell membrane pretty yellow. And the centrioles, it's always been a centriole. And now the chromosomes are now chromatin, E2 chromatin. So it went from chromatids to chromosomes to chromatin. And let's go ahead and color that nucleus. Nuclear membrane. And F2 chromatin. So like I said, it went from chromatids to chromosomes to chromatin. And we're done. So much prettier when it's all colored in. Mitosis, yeah. Cell division. See you later, guys. Subscribe.